Okay, so the next type of macromolecule that we're going to look at are proteins. Proteins are composed of carbon, hydrogen, oxygen, and nitrogen. So nitrogen is the new addition to this mix. They can also contain sulfur, one particular amino acid. They're also called peptides or polypeptides. Their roles in the body are by far the most diverse of the three classes of macromolecules, with nucleic acids being sort of the most specialized, and then lipids and carbohydrates. So they can act as enzymes, which are biological catalysts, and we'll talk far more about this a little later on in the unit and next unit. They have structural roles. So, for example, actin and myosin are microfilaments that are responsible for, like they're your muscle fibers. They're responsible for the cytoskeleton structure and so on. We can have protein hormones. So insulin, for example, antibodies are part of your immune system that helps fight disease. So they're involved in your immune response and immune memory. And they can have a role in transport through the cell membrane. And that's really only a small fraction of the roles that they could take on. Monomers are amino acids. And they're called amino acid because they contain a central carbon atom bonded to an acid group, so a carboxyl or organic acid group, and an amine group, as well as a hydrogen. And then the fourth substituent, the fourth component on that central carbon atom is an R group or variable side chains. And all amino acids are going to be at least a little bit polar because of the acid group, which is very polar, and the CN and NH bonds are also polar. But some are more polar than others, and that has to do with their R group. So here that is visually. Here's my amino group. Here's my acid group. I have a hydrogen, and then this R group is going to vary. So in theory, there's thousands, millions of different possibilities for what this R group could be, but practically speaking, only 20 amino acids are of biological importance. So our body has the ability to make proteins out of only 20 of them. The other thing I want to point out is that this hydrogen here, sometimes you'll see it written on the amino group, so you'll see it written as an NH3. If this is an NH3+, then there is no H shown on the carboxylic acid group. You might also see this H attached to this O. In this case, it would be the C double bond O, OH, and then an NH2. They're both the same molecule. It depends on the pH of the solution that it's in, whether that H is stuck to the O or whether it's stuck to the N. And these are the 20 biologically important amino acids. They're divided into 4 or 5 different classes, depending on what you're looking at. But the big ones are nonpolar, polar, and then charged. And sometimes you'll see the addition of this aromatic as well. Sometimes you'll see these lumped in other places. So the nonpolar amino acids would be glycine, and here's the R group highlighted in yellow. So glycine's R group is the simplest possible R group. It's just another hydrogen. Alanine is a methyl group, a CH3. Valine, again, a very simple hydrocarbon chain. Leucine, isoleucine. So leucine and isoleucine, you can see they'd have the same chemical formula. They're just rearranged. It's which carbon this methyl group is stuck to. Methionine, that's one that has sulfur in it. And proline is kind of the coolest, weirdest amino acid. You can see it actually has a cyclic ring in it, in its structure. So that's when this appears in proteins, it tends to cause a kink or a bend in the protein structure. We have polar amino acids, serine, cysteine, and threonine, and they're polar because here we've got an OH group, here we've got an SH group, here we've got an OH group. The aromatic amino acids are called aromatics because they contain a benzene ring, and they do tend to be stinky. So things with benzene rings, they're called aromatic compounds because they're aromatic, they're stinky. So here, phenylalanine, tyrosine, and tryptophan. So you should be able to see why these get multiple classifications, because they do all have aromatic rings in them, but they also could be polar or they could be nonpolar. So phenylalanine is nonpolar. Tyrosine and tryptophan will both be slightly polar. Tyrosine because it's got this OH group, and tryptophan because it's got this NH group. Then we've got our basic amino acids. So histidine, lysine, and arginine, they both have these NH2 groups, and you'll see they're slightly positive. So these in general are considered charged. They'll be slightly positive because they're capable of acting as a proton acceptor, as an H plus acceptor. And then we have acidic amino acids and amide derivatives, and all of these really are going to have some sort of acid, organic acid, or amide group. You don't need to memorize these. Oh my God. I think I had to memorize them in biochemistry. But it was more, I had to be able to recognize them. I don't think I ever had to draw one from scratch, but I had to recognize roughly what they were. The big reason you need to be able to do that is you need to understand whether they're polar, whether they're nonpolar, and what that's going to do to the structure of a protein. And we'll talk about that a little later. So we can also kind of classify these by their dietary requirements. And different organisms have different amino acid requirements. For humans, 12 are what we call non-essential. So we can make them from other amino acids in our body. And 8 are considered essential. So we can't manufacture them. We have to get them from our diet. And this is why really we have to eat meat or be very careful vegetarians or vegans. So for example, rice alone doesn't contain all 8 of these amino acids. Beans alone don't contain all 8 of those amino acids. But if you eat rice and beans in combination, they provide the all of the 8 that you're missing. The bond that holds proteins together is called a peptide bond. It forms between the carboxyl group of one amino acid and the amine group of another. And same as the previous examples, what we need to do is figure out how to get rid of the water. So here we've got an O on this compound and H's on this one. In some cases, if the pH was different and you had an OH here, then you would only be taking one H from this nitrogen. You're going to have the O and the 2 H's combine to release H2O, and a bond forming between the N and the C. So you've got a C double bond O, N. That's called a peptide linkage. If you've taken grade 12 chem, we'd also call that an amide. Now, since there are 20 different amino acids, this makes for millions of different possible proteins, because for each amino acid or for each spot in a protein, you have 20 different possibilities. So for a protein that's 20 amino acids long, there's 20 to the power of 50 different possible sequences. And the sequence is important because the sequence determines the 3D structure. So we talked about whether things are polar, nonpolar, and 3D shape. That becomes important because the sequence of amino acids will determine through their interactions the 3-dimensional structure. So the first level of 3-dimensional structure that we have are the secondary structure. The sequence itself is what we call primary structure. The sequence begins folding into smaller substructures that we call secondary structures. So this is a small substructure called an alpha helix. You can see it's kind of twisted like so. And that would fold due to hydrophobic interactions, hydrogen bonds, dipole-dipole interactions between the R groups of these amino acids. And these small substructures then combine to form a whole 3D structure. So you can see this is a molecule, it looks like a hemoglobin chain. This is a molecule that is composed primarily of these alpha helixes with some loops in between them. And so the whole 3D structure of the whole protein consists of 1, 2, 3, 4, 5, 6 alpha helixes. All right. So secondary structure is a small sort of substructure of the whole greater structure. It's sort of like one whole condo building. is made up of multiple different condo units, and each unit can be made up of different rooms, if that helps. So the forces that maintain the structure are hydrogen bonding, which we talked about earlier, van der Waals forces or nonpolar interactions, covalent bonds in the form of disulfide bridges, covalent bonds that hold the chain itself together. You can also have some ion dipole forces and a hydrophobic effect. So the protein will curl so that any hydrophobic regions are tucked away from water. So this is an example showing these different substructures. This is sort of the loop or fold of the protein, and you can see our R groups sticking into and out of the main chain. So here we've got some dispersion forces holding these nonpolar regions together. So this would be a nonpolar interaction. Here we've got some hydrogen bonds. In this case, that's holding these two chunks of the molecule itself together. We can also have hydrogen bonding to water, or this is an example of an ion dipole bond between, this would be some sort of amide, or sorry, some sort of amine, and then a water molecule. Here's more H bonding outside to water. This is a disulfide bridge. So here you've got two sulfurs, and what can actually happen is if you've got two sulfurs in a cysteine molecule, if they cozy up close enough together, they'll react and get joined. This is actually what gets changed if you have a perm. So in your hair, disulfide bridges hold the protein fibers together. What you do when you get a perm is you break those disulfide bridges, put the hair around a curler, reform the disulfide bridges, and the new disulfide bridges mean that your hair is now curly and will stay that way because it's a permanent change. And we already talked about the levels of protein structures. So the simple 3D structures are the secondary. So the alpha helix was the example you saw, the beta pleated sheet. Tertiary structure is the whole three-dimensional structure and its interactions between R groups and so on. Some of it happens naturally. So the secondary structure occurs naturally. The tertiary structure, in some cases, may occur totally naturally. In other cases, it may need molecular chaperones. So it may need other proteins to actually help it fold. And this is important because if something happens and alters the structure, then it can't refold naturally. Once you've kind of messed with it, you've messed with it. And some proteins, but not all, have what we call a quaternary structure. So a quaternary structure is sort of a higher 3D level structure where multiple peptides might have to come together to make one functional enzyme, or it could require a cofactor or a coenzyme. So for example, hemoglobin has a tertiary structure all on its own as a hemoglobin chain, but then you have to add a central unit called a heme. So if I go back to this slide, it would be something like this. So you've got this additional molecule that's forced to sit in there with an iron in the center of it, and that's where the oxygen is going to bind. So the hemoglobin protein itself would have a tertiary structure, but the presence of that other group gives you the quaternary structure. If we expose proteins to significant changes like extremes of pH or temperature, then the bonds that hold the proteins together can be broken. So unlike a covalent bond, those intermolecular forces are a lot weaker. It doesn't take a lot of energy to disrupt the intermolecular forces. So if you add a little bit of heat, you're going to disrupt the intermolecular forces, and the protein is going to lose its 3D shape. Once you lose the 3D shape, you'll also lose its function, and that is a process called denaturation. Here's an example of the primary structure of a protein. And that's it for this one.